Hi, this is Lila
and Harlow,
and here's what we learned about esophageal cancer and cell communication as well as the cell cycle during this Unit 4 project. So esophageal cancer relies greatly on the mitochondria in the nucleus as key organelles for regulating the growth and proliferation of esophageal cancers because the mitochondria is responsible for the metabolism and the nucleus is responsible for DNA and DNA replication, which are key parts in it dividing.
How do cells communicate with each other with this cancer? They communicate like most other cells. They use ligands and then signal receptors. The thing is, they use like growth factors that cause other cells to become cancerous as well, and it can spread the cancer throughout the body.
Yeah.
And then, what are the components of signal transduction pathway and their individual roles? The components are ligands, receptors, second messengers, and responding molecules. Ligands are the signal itself. They connect to receptors on other cells that then tell the cell to do certain things. Second messengers are either sent out or amplify the signal inside the cell itself and then activate another molecule. And then the response molecules,
So the environment is really key in eliciting these cellular responses because cells respond to environmental signals through networks of receptors and signaling molecules and transduction pathways, as previously mentioned. And these allow organisms to adapt to their surroundings based off the environment triggering these pathways. Also, the structure of a molecule greatly impacts its ability to pass through the membrane, which can be important when talking about how signals are received based on whether or not the signal is going to be received extracellularly or intercellularly. And things that impact this are the polarity, the size, as well as the charge and the composition of the membrane, whether or not it's going to allow different types of molecules to pass through. And the cellular response of a signal transduction pathway is a very highly regulated response due to the specificity of cellular pathways, and these cellular responses are the final result in cell communication, and they are more of the measurable result in the response to the initial signal that is observed by the cell to help the cell react to whatever is happening and maintain homeostasis. Also, the structure of a signaling molecule impacts the activity of a signaling pathway because the molecular structure of signaling molecules helps us understand how the pathway will work because it determines how a molecule will bind to receptors and that impacts the strength, specificity, and duration of a signal transduction pathway. And these properties include polarity, size, functional groups, and flexibility to conform to the receptors, as well as chemical stability.
Next is the events of the cell cycle and mitosis. During the cell cycle, main part of the cell cycle is interphase. Cells spend about 90% of their time in interphase. Phase 1 is G1, where the cell grows in size. It makes sure it has enough resources to divide, and also if there is room to divide and the cell needs to divide. And then is the S phase, where the DNA gets replicated. Then is G2, where the cell grows bigger and makes sure there are like no problems with the DNA at all. And and then the cell goes through mitosis. The first part of mitosis is prophase, where the nuclear envelope breaks down and DNA starts to condense into sister chromatids and chromosomes. Next is metaphase. Metaphase, the chromosomes move into the middle of the cell in a line. Spindle fibers connect to the chromosomes from centromeres at the poles of the cell. The spindle checkpoint happens here, where make sure all the spindle fibers are connected to the next is anaphase. The spindle fibers begin to pull the sister chromatids apart away from each other. Lastly, telophase occurs when sister chromatids are fully separated from each other. And then cytokinesis, split, 2 daughter cells. Next is what is the role of checkpoints in regulating the cell cycle? The role of checkpoints is to make sure that the cell is working correctly and that it can divide, should divide, and like the DNA is all good and it has enough resources. At G1, if there's anything wrong with the cell, it goes to G0, where it either fixes the DNA or gathers more resources, so it can divide later on. Later checkpoints in the cycle, though, if like anything is wrong, like the DNA is damaged or that like there's not enough resources, the cell either desperately tries to fix those or commits apoptosis. And
yeah, for G2 and M checkpoints, it goes to apoptosis if it fails.
Also, the regulation of cell cycle disrupted to cause esophageal cancer. Cancer is the unregulated replication of cells. In most cancer cases, including esophageal cancer, it's caused by mutated genes. The genes that code for proteins that help checkpoints in the cell cycle are affected, so the cell like can't check itself that there is no problems, and it just starts like rapidly dividing because it can't like check itself and just keeps going. The mutated cells multiply and lead to more mutated cells and tumors. The mutations can be caused by genetics or use of substances like tobacco and alcohol, as well as other risk factors.
That's what we learned about esophageal cancer and cellular communication and the cell cycle. Yay.
Yay.